spices as natural antioxidants, applications in food and health implications. Audio recorded August 31, 2023 by Sybil Mann Baylor. For educational sharing in audiobook Flor de Aromatic Cosa Nova. Welcome to this channel. Like and share. Jorge Hendal** Sao Paulo State University, UNDSP, Institute of Biosciences, Letters and Exact Sciences, Department of Engineering and Food Technology. Abstract. The growing interest in replacing synthetic antioxidants with natural ones in foods has fostered research on plant sources, characterization of raw materials and identification of new antioxidant compounds. Oxidation reactions are not an exclusive concern of the food industries. Their study is also widely needed to avoid undesirable implications on human health. The purpose of this review, after presenting general aspects of oxidation, is to focus on the benefits of using spices as natural antioxidants, especially those of the Labiatae family, widely used in Brazilian cuisine. Keywords: oxidation, natural antioxidants, spices. Lipid oxidation that occurs in food products is one of the main concerns in food technology. It is responsible for unpleasant odors and flavors in products, with a consequent decrease in nutritional safety and quality, caused by the formation of potentially toxic compounds. Prevention is economically important and fundamental for the protection of human health. Tsai et al. 2005. Antioxidants are presented as an alternative to prevent oxidative deterioration of food and minimize oxidative damage in living beings. As the use of synthetic antioxidants in the food industry has been the subject of questions regarding their innocuousness, demonstrating the possibility that these antioxidants have some toxicity. Bauer et al. 2001 research is focused on the search for natural compounds that exhibit this toxicity. Functional Property Memo received for publication on October 24, 2010, accepted for publication on February 4, 2012. Engel, 2002. The very demand of the consumer reinforces the potential market for natural antioxidants, since there is great discomfort on the part of the population in maintaining the acquisition and consumption of industrialized foods, the production of which is known to involve the use of substantial amounts of synthetic additives. Landau et al. 2002. Hamerowich et al. 2004. The beginning of the new millennium, with the increase in life expectancy and the growing information disclosed about health, lead the consumer to seek more and more a healthy diet for the prevention of diseases. At the same time, advances in science allow for better knowledge of the properties of foods. Several culinary herbs and spices have already been reported to have antioxidant activities, even suggesting potential benefits to human health. Yanishleva et al. 2006. Viodemartos et al. 2011. Despite the growing biochemical, clinical and epidemiological evidence for many of the functional compounds, there is still no established scientific consensus that supports decisions within the scope of more global public health policies regarding their use. There is a need, therefore, to deepen the field of research related to the identification of these compounds and their mechanisms of biological action and the elaboration of foods aimed at special health purposes. These considerations point to the importance of studying spices as natural antioxidants, in order to minimize oxidative processes and prevent metabolic changes in the human body, a topic that will be the subject of this review. Oxidation and production of free radicals. Lipid oxidation is a complex phenomenon induced by oxygen in the presence of catalysts such as heat, free radicals, light, pigments, and metal ions. Labiatae et al. 2007. The oxidation mechanism can occur in animal and vegetable tissues and in products obtained from them, such as oils and fats. There are three ways in which oils can undergo oxidation: autooxidation, photooxidation, and thermoxidation. Berger and Hamilton, 1995. All these routes contribute to decrease the quality and modify the structure due to the degradation of liposoluble vitamins and essential fatty acids, in addition to affecting the integrity and safety of foods through the formation of potentially toxic compounds. Silva et al. 1999. Autooxidation, a complex reaction catalyzed by extrinsic factors, begins with the formation of free radicals and occurs mainly in unsaturated fatty acids, due to the electronic instability that the double bonds present, making the hydrogens attached to the carbons, where the unsaturations are located, easier to be removed from the carbon chain. 1996. Salem Jr. 1999. According to Gordon 2001, the autooxidation of oil occurs through a chain process that involves a sequence of reactions, from the formation of the free radical under conditions favored by light and heat initiation phase, formation of peroxides and hydroperoxides propagation phase, and formation of stable oxidation products such as aldehydes and ketones termination phase. Photooxidation is another proposed mechanism for the oxidative degradation of oils, considering that the direct reaction of lipid substrates with atmospheric oxygen is thermodynamically unlikely to occur. Gordon, 1990. The reaction is promoted essentially by UV radiation in presence of sensitizers, involving the participation of singlet oxygen as a reactive intermediate. 1996. To simulate the process in which oils are subjected to high temperatures and to study the compounds originated during the thermal stress of oils, the use of thermoxidation is very common. Method that consists of subjecting oils and fats to high temperatures, but without the presence of food, that is, without moisture and other components that come from food. Therefore, temperature and oxygen from the air are the main variables that must be taken into account. The oxidation of oils in the absence of food is also used to study the efficiency of antioxidants against the formation of degradative compounds harmful to human health. Labiatae et al. 2005. The hydroperoxides formed during the lipid oxidation process are essentially odorless. However, they decompose into a wide variety of volatile and non-volatile secondary compounds. Warner, 2002. The volatile compounds are partially eliminated in oil heating processes, and their importance is related to the sensory characteristics. Jorge, 2004. Non-volatile degradation products, on the other hand, are of great interest from a nutritional point of view, since they are part of the diet when they remain dissolved in oil and are, therefore, incorporated into foods. Warner, 2002. Studies reveal that highly or moderately oxidized foods are quite atherogenic and that oxidized cholesterol appears to be particularly dangerous. Its oxidation products, called cholesterol oxides, which comprise a vast number of compounds, promote atherosclerosis, in addition to mutagenic, cytogenic, and arterial damage. Deco et al. 1998. Joe 2004 states that the oxides affect the activity of several enzymes, alter low-density lipoproteins LDL that are involved in the development of arteriosclerotic lesions. It is believed that LDL oxidation is the main cause of cardiovascular diseases, and the decomposition of peroxides formed by the action of lipoxygenase may be the initial mechanism of LDL oxidation. Hydroperoxides and products can also react with proteins, altering the nutritional value, rheological properties, and sensory characteristics of foods. Free amino acids, mainly methionine, cysteine, histidine, lysine, and other amino acid residues undergo substantial oxidation in the presence of free radicals. Jacobi et al. 1995. As a consequence, there is a decrease in the digestibility and bioavailability of essential amino acids, a reduction in protein solubility, and changes in the texture and color of foods. Kirk, 1984, Jacobi et al. 1995, Ferrari, 1998. In the human organism, the oxidation of biomolecules and the production of free radicals are integral parts of metabolism. Free radicals are unstable molecules because they have an odd number of electrons. To achieve stability, these molecules need to acquire electrons and therefore react with most neighboring compounds, oxidizing them. Sanchez Moreno, 2002. Reactive oxygen, Roscommon, and nitrogen RNS species are continuously produced in the body and, at physiological concentrations, perform essential functions related to cell cycle signaling. Immune defense. Ribeiro et al. 2005, and reproductive biology. Sonaka and Corpus, 2004. However, in supraphysiological concentrations and deficiency in the protective system, there is an imbalance between aggressors and defenders, characterizing oxidative stress. The generation of reactive oxygen species Roscommon is inevitable in the organism's aerobic